everybody. Welcome to Ask the Experts. We look a little bit like the Brady Bunch, but I'm glad we're all here. So this is your chance to have a conversation with some of the industry leaders in the field of transfusion medicine. So let me begin by introducing everybody. I'm going to moderate. I'm Dr. Barbara Bryant. I'm a transfusion medicine physician, and I'm an adjunct professor at the University of Texas Medical Branch in Galveston. Next, we have Dr. Lou Katz, adjunct professor of internal medicine and infectious disease at Carver College of Medicine at the University of Iowa. Dr. Katz has over 30 years career in infectious disease, HIV medicine, hospital epidemiology, and public health. Next, we have Dr. Art Bracey, professor of pathology and immunology and the chief of clinical pathology at Baylor St. Luke's Medical Center in Houston, Texas. We have Dr. Alice Chen, who is a transfusion medicine physician and appears to be on call. And she is with Gulf Coast Pathology Associates based in Houston, Texas as well. And then we have Dr. Chester Ngioski. He is an assistant professor of anatomical and clinical pathology, and he's at Tufts University School of Medicine. So we have received some submitted questions, which we will address. But if you have questions, please just put them into the chat and we will go through those. So let's get started. So first question up. As a hospital-based donor center, if we manufacture platelets using the 48-hour method, will we be required to do Verax testing at any time? All right, who wants to take that one? Lou.
Oh, that's an Art question, I think.
Absolutely.
Well, no, if you do the high volume delayed 48 hours, you get a 7-day out date and you shouldn't have to do anything else.
Okay.
But you have to hold it for whatever the time frame is in the guidance, 12 or 24 hours after inoculating. I can't remember, but that's what we're doing. And our hospitals are very happy because they don't have to do anything else. They don't have to do anything but take it out of the shaker and send it to the bedside.
All right. All right. Let me move on to the next one. How soon should we expect a 7-day PRT platelet to be made available? Who wants to take that one?
Well, I would say that's up for grabs right now because that's got to go through the FDA process, and who knows when the answer.
Yeah. Ask Cirrus.
Just don't know.
Okay. Anybody have any
it's a
idea?
Container issue, and they need to get their container approved for 7-day storage.
Any idea where they are in the process? I know everything kind of came to a halt with COVID-19, but everything's.
I stopped asking about the 20th time I got, we're working on it.
Yeah.
So I'm sure that somebody from Cirrus will be available to answer the question, but it's a big issue because we make intercept platelets at my center, and the hospitals would really like to have the same out date for everything.
Yeah, I got to say one thing for sure is that looking at avoiding platelet wastage, 7 days is a gem. It's almost necessary.
Yeah.
Very good. One of the members at the meeting said, how do you see the development and manufactured red cells and platelets over the next 5 to 10 years?
I just downloaded a chapter on this issue this morning, and I haven't read it. So if somebody wants to talk to me on Monday, I'll have a better idea. People are working on it, but the regulatory hurdles appropriately are really incredible. I mean, you're going to use farmed red cells, you better know they're safe. Not to mention that they work. But I think in 5 years, we won't have them.
So the one thing that we're working on right now is we're working on the trial with the Red-S trial, with the Cirrus treated RBCs. It's kind of slow in coming, but the issue, of course, is looking at neoantigens and new incompatibilities, but there will be some trial data coming out. There's some already from from Germany and some from Greece, but the trial is in process in the States. but the notion that Lou talks about, put some cells in a vat and grow them. The only question there is really, it's you can do it, but is it going to be, really economically, would it make sense economically?
Yep.
So reaching back to my PhD days, I actually worked on hematopoiesis and grew blood cells in culture. But when you're talking about a transfusible size volume, you are talking about ginormous containers of a very expensive growth media. So that's, I think, what Dr. Bracey is alluding to in terms of cost effectiveness. But honestly, there's just some flat out logistic aspects because it's very, very literally large volumes. So you're talking about like rooms full to generate a couple of units. I don't know the exact translation of size, but it's a very large scale effort to generate a fairly small amount of transfusible product.
Yeah, I think the research efforts to date grow billions of cells. It's just not close, and you got to figure out how to scale it. And then, is it worth it? Are the risks and availability of the current real product sufficiently threatened that it will ever be worth it on a risk-benefit standpoint?
I think from my perspective on this question, looking back over my almost 4 decades in transfusion medicine, especially with my time actually down in Texas when I was in the Air Force down there, I remember a colleague approaching me and saying, "Chet, you're going to be out of business in a few years because we're going to be having artificial blood." And that was like 30 some odd years ago. And in seeing what's happened in that time interval, I mean, I am enthusiastic for developing these types of assets. As everybody here has mentioned, there are pluses and minuses. I know when I got up here at the current hospital that I'm at, Bay State, we actually were involved in discussions with some folks in Boston through the medical advisory committee of our regional Red Cross and the Mass Medical Society, looking at some of these products. And the upshot of the whole thing that I saw was, although I could see that these would have special applicability, like, for example, in trauma settings, especially in the military, maybe with people who didn't want to take blood products. I mean, the bottom line from my perspective is I don't see us substituting for the real thing. And the importance of having, blood collection capabilities and people to donate blood, I don't see that being supplanted, to be honest with you.
And then, the other thing is that as patient blood management comes along, it's not going to eliminate red cell transfusion by any means, but it's already had an impact on demand. And as surgical procedures get, smarter and perhaps less invasive, the need may diminish so that the supply clearly will be adequate.
I'm getting several other questions along this line about if we manufacture red cells and they have to be more cost effective, but there'd also be a need to engineer some rare phenotypes. That would be very interesting.
Yeah, that would be. There's a biology point here too, which is that what cell are you going to start with, right? Because if you start with the hematopoietic stem cell, that's actually a lot more factors to push it down towards red cell. And nobody's particularly interested in generating a whole bunch of lymphocytes. So if you start with a hematopoietic stem cell, then you have kind of infinite growth. But if you have to start further downstream, you have more limited growth. And I don't know. I sometimes think about at what point are you going to start that growth pathway? You can't you can't reproduce a completed erythrocyte. You have to have a cellular, further upstream progenitor to be able to grow it. So where do you start, and what phenotypes are you going to generate from that?
Well, then the other thing is that if you look at what's been going on with the rare donor registries and the use of molecular type testing to screen donor populations, you know, we've been able to find more rare folks now that we're better at meeting certain rare donor needs now than we ever were. So, you know, another strategy to deal with the rare donor situation is to be able to find them in a smarter way.
Yeah, it's an IT solution that is very important to the rare donor issue, is that knowing that I have a Bombay donor in Iowa would be an important priority when somebody needs it in the middle of the night.
Mm-hmm.
Well, that's what the military essentially does, but just not with rare donors, right?
Yep.
And actually, that gets to an important, so there's this group now that's responding to section 209, question number 3. There's Congress wants to know how to, you know, manage during an inventory challenge. One of the things that we don't have in the U.S. is a way to actually survey efficiently all of the inventory. We were really good about blood centers. We know what we have in the blood centers, but then it's in the hospitals. And the only reason that we don't know, I think, is pretty weak, because we don't have a national blood policy. So, you know, we could link all these things up, and like Lou said, if he's got a Bombay in his place, the blood centers can find that now, but really trying to link in the hospitals too, I think would be great.
Well, the Advisory Committee on Blood and Tissue Safety and Availability had a 2-day meeting this week. And one of the recommendations that's clearly going to come out of that meeting is let's get a national blood data infrastructure so we know what's where.
All right, that's what we need.
Yeah. How detailed would that be, though?
I think that's to be decided, and I'm certainly of a mind to say let's start with the critical stuff and not worry about where the pooled cryo sits. Let's know where red cells and platelets are, hospital and blood center, ABORH, and start there and build from there.
All right, let's see. Whatever happened to attempts to create a synthetic erythropoietin?
I have no idea.
I haven't heard either.
Now you'll get coffee now?
So the erythropoiesis that we're struggling with right now, and I had a case today. So, you know, this patient's got an LVAD, and he's 7 point whatever, and they're trying to give him iron, and he's not going anywhere. And so questions about, you know, treating the anemia, and can you treat the anemia in a cost-effective and safe way? So rather than just can you know, make the blood in a bag, what about making the blood in the patient?
Exactly.
Yeah,
that's right. Yeah.
Although that goes back to what the question was asked. You know, if you have a synthetic erythropoietin that's a little cheaper than what's currently available, then and has a nice safety profile that isn't going to cause thrombosis, then could you help them along? Especially these patients that have renal issues, so you know their EPO levels are not quite normal.
Right.
But they're not really end-stage renal, so you can't justify giving them full-blown EPO.
Right.
All right, I've been asked this question 3 times in different ways, but bottom line, will there be a deferral period for donors who may participate in the vaccine trials for COVID-19, and then moving forward, once the vaccine's available? Well, so that's when it's an investigational vaccine, but then afterwards, will there be a deferral? Do we have any idea?
Well, AABB is actively engaged OBRR, CBER OBRR at FDA in this question, and FDA's initial discussions are very permissive, that particularly if it's not a live virus vaccine that we think might cause trouble, I think they're going to leave that decision up to blood center medical directors, collection facility medical directors. If it's a kill vaccine or a non-replicating vaccine, my understanding is that we're going to have discretion to accept those donors. And that comports with the UDHQ at this point, where live virus vaccines are looked at one way and non-replicating vaccines another. So I think it's going to be permissive.
Okay, very good. I have a question about.
Wait, but before you leave that,
sure.
So what was the stance on these gene therapy vaccines that aren't live and are not?
I think they're being looked at like the non-replicating vaccines.
But they're not really, right? Because they are. They're integrating and then.
But there's no production of intact virion.
Viral particles?
Yeah.
Okay.
Hey, Lou, do you think there's going to be more than one vaccine that's going to get the nod?
God, I get to answer a question with a high level of certainty. Yes. Yeah, I mean, there's too many for only one to make it. But there's some.
And I've read too that if only one makes it, you're not going to be able to ramp up production to sufficient levels for one. So
one of the reasons for all the parallel.
Yeah, I think another interesting question vis-a-vis the vaccine issue has to deal with the convalescent plasma donors and whether or not they would be candidates for getting the vaccine, either as a patient, in follow-up there, or if they recover and they come into the program to donate, how that would affect them. Any thoughts?
Well, so the convalescent donors are currently deferred for 3 months, and we're seeing in our program that when we get our donors out to 3 months, which we have some now, their levels are starting to drop off. So my guess is that COVID patients who got convalescent plasma are not going to be a robust source of the product later on down the line. We'll see as time goes on. And the question about vaccines is going to be, do you get a robust enough response of the right immune factors that you're going to want to use it? And again, let's see what the clinical trials show us.
Along the line with the COVID-19 vaccine, who do you think will get it first, second, on down the line? And what are your opinions on the distribution?
Martin and Alice will get it first.
1600 Pennsylvania.
No, I mean, healthcare workers at the front line, I think, will be the first to get it. And I think that will, I think that's appropriate. They won't give it to me because I don't go to the hospital.
I think they're probably going to go for like nursing homes or something like that. It's the mortality. The frontline workers are getting a lot of exposures, but the mortality in places like nursing homes is much higher.
Well, as one who just turned 70, I hope that they just start with the old guys first.
Yeah. All right. So there was a talk yesterday about cold stored platelets. Do you think that they will be available for civilian use anytime soon?
I'm telling you, he got me excited. I want some.
Well, there's already one place that I think Mayo got a variance to allow to the use for trauma support. But I think they're probably the only place right now. But so it's interesting. So, as a hospital that takes care of people that are bleeding acutely, I would certainly like to try to use them in a certain set, not in a prophylactic transfusions, but clearly in the therapeutic ones.
And in January or February, I got the validation plan from a blood center that is making cold platelets that the FDA likes, and without COVID, we would have submitted them. Our trauma people would love to have them, but COVID got in the way.
Yeah.
Yeah, I'm definitely interested in for our hospital system here.
I mean, 14-day shelf life, you talk about, even if you don't get the 21-day, 14-day shelf life, you talk about 7 days helping. 14 is going to even be better, right?
Some of y'all have been doing this long enough that you remember when we kept them in the refrigerator, but it was only for 3 days back then. I remember when I was first started as a medical technologist, when you need platelets, you had to check the dates on the ones in the fridge versus the ones that newfangled way of keeping them at room temperature. So that was kind of really interesting. So, okay. And then there's some studies being done on frozen platelets and along those lines. So, okay. And somebody, Brian Nobles, put through a comment that San Antonio, so I believe San Antonio is also using some of the cold platelets. So,
well, I won't say where I got my validation plan, but it might have been San Antonio.
It might have been. Thank you, San Antonio. All right. Let's see here. I have a question about what is your experience with intercept platelets in neonates?
Well, we don't have kids, so I'm zip.
What do y'all think?
We make them, but the hospitals that are taking them don't use them in neonates. Okay. So I have none.
Okay.
I know from Lisa Hench over at Texas Children's that they've been interested, but I don't know if it was specifically for neonates. Next time we're going to have to get a PD blood banker on here.
Yeah. Mm. Lisa Hench, you can go ahead and text me. I'll tell everybody what you said. I also just got a message from Lisa Fultz saying that South Texas Blood and Tissue Center has licensed cold store platelets for 14 days for actively bleeding patients only.
Yep.
So, okay, all right.
You want to ship them to Houston?
They probably would.
They probably would.
We know some people, so there you go. Let's see. Let's see. Here's one about donors. What's the appropriate deferral period for an excised melanoma in situ? What are y'all using for your deferral period?
Did you say melanoma in situ?
Yes.
I mean, if it's completely removed, I can't imagine that it would be.
Yeah, healed, stitches out.
Healed stitches.
I think that's what we do here.
Okay. All right, let's see. I've heard from Lisa Hench.
Oh, no way. Go, Lisa.
She actually has this up on an 82-inch TV because she says the panel is too big. So I'll show that to everybody. But she says they don't use them. They were helping to validate these platelets, but they're not using them for neonates. Thank you, Lisa. All right. Oh, San Antonio said they'll send them to Houston. Okay.
This is really live. This is awesome.
Hi, great. This is tons of fun. Okay, let's see here. Mary Berg from Colorado, University of Colorado in Denver, says that they use them in neonates and they have not had any problems. So that's good to know. That's really good to know. All right. Let's see here. I got all these messages coming through. All right. Next one up. If the blood center is collecting CCP more frequently than every 28 days and the COVID antibody level diminishes to negative, can you convert those units already collected into regular apheresis plasma and make it for transfusion?
We do.
Even if they've been collected more frequently than the 28 days?
We don't do it. I mean, we test antibody levels on every donation. So if they've given several times and now their level has gone below our cutoff, we convert it to transfusible plasma and then ask them to lay out of the program. So it would be once.
Okay. All right.
We've been doing it. If it's not legal, somebody should tell me.
Just out of curiosity, have you ever seen someone's titers drop off but then come back up?
Well, once they're negative by our criteria, we just arbitrarily decided that's enough. We do them weekly for a month, and then we'll do them 2 weekly. But if they go negative, then we say thank you and you're a hero. And if you want to be a donor, we'll arrange it. But we don't let them back in the CCP program.
So I've got a question about the, you know, the titering, et cetera. It's all rather vague now. And there were some recommendations that came out with one instrument. I guess it's the Vitros. Well, tell me a little bit about what you do with the antibodies. What's your definition of a solid CCP donor?
Well, our program has required a positive PCR for diagnosis.
Yeah.
Then we have used the Abbott Architect to screen. That's a nucleoprotein assay.
Yeah.
And originally Euroimmune and now Diasorin, which are spike assays. So they have to have a positive PCR and be positive on both antibody assays. And we use a cutoff in the Diasorin, I think it's 4. And we managed to qualify, because we require a positive PCR, the vast majority of our donors are antibody positive. And most of them make the threshold on the Diasorin. We'll be switching to the Vitros as soon as we can, and then we'll use 12. And my understanding is the FDA says you didn't use ortho-Vitros for what you've got in inventory. You have to call that low titer. And that's fine. It's all we have until we're up on the ortho-Vitros, and the hospitals are fine with that.
Oh, okay.
So I have a question, because at one point I kind of dug into more of the titer aspect of these antibody assays, and they're all labeled as semi-quantitative as opposed to truly quantitative. So how is FDA taking that? I mean, I haven't gotten a clear vibe on
the only assay approved for CCP now, the label at high titer, is ortho-Vitros IgG. That's the only one. You have to have 12 or up. SCO have 12 or up for that. The others don't have semi-quantitative claims. We chose our thresholds because there were unpublished but acceptable small amounts of data that said neutralization titers with the Euroimmune IgG assay, IgG ratio of 4 or up was associated R squared equals 0.5 approximately, with a neutralization titer above 200. So that's what we used originally. And now we're going to switch over to ortho-Vitros and use it as the FDA has said. They've told us very clearly we don't have to throw away what we've got in the freezer. But the people at the hospital need to know it's not high titer according to the EUA. And the other thing that they've told us very clearly is we're not allowed to give titers to patients or doctors, because the assays are not approved as quantitative assays. And I get it. I think it's a little stringent, but so I'm choosing to interpret everything simply and asking forgiveness, not permission, if I'm doing something wrong. I mean, we just can't interrupt the flow right now.
So you have an innovative treater who calls for 3 liters because he's going to do a plasma exchange. Honestly, so how are you guys handling requests for greater than N of 1?
Our recommendation, and it is exactly that to our 119 hospitals, is 70 kilos or less. Start with a unit and see what happens. Above 70 kilos, give 2, and we don't want more than that, because the way that the data that we're using has been generated is essentially with 1 or 2 units, right. And we need to have it available for more patients than yours. So I would probably get pretty involved in a request for 3 liters, as in no.
Right. No, so yeah, it's interesting because what we've done also is, under 70. Well, actually, our cut point is 100 kgs. After 100, we get 1 and 2. But
so we haven't received a request like that, but I have heard that come up in some other discussion groups that I've participated in. The issue of using therapeutic plasma exchange, for example, to help treat the cytokine storm and then use convalescent plasma. I mean, if I was confronted with that, and we haven't been, thankfully, the consideration that I would bring to bear here is not just for that individual patient, but clearly for the inventory, as has been discussed. And perhaps a strategy that I might employ for that patient would be to do a therapeutic plasma exchange. And in the replacement fluid mix, to consider using regular FFP and or albumin for the bulk of it. And then towards the end of that, add on 2 or 3 units of convalescent plasma, depending on how our inventory looked like. With the idea that what we're trying to do here is to debulk, if you will, immune mediators from the cytokine storm, and then replenish the patient with protective antibody, as well as potentially regulatory molecules that might be in the CCP that could help them. This is sort of like a strategy that we've used with some of our TTP patients who have severe sensitivities to FFP as we go on. And it allows us, I think, to enjoy the benefits of both the depleting type of process with the apheresis and then replenishing it as well. Those would be my thoughts.
Well, and the other thing I'd consider is if you're dealing with cytokine storm well into the second week, usually, and beyond, and I'd want to know they were antibody negative, the recipient was antibody negative before I even talked to them about large volumes of convalescent plasma. I think we all know the Dutch stopped an RCT because they found out that most of their severely ill patients that were candidates under the criteria of the original trial were already antibody positive, including a high rate of neutralizing antibody. And I don't see that using convalescent plasma is going to be highly likely to have a lot of benefit. There may be some innate immune things and anti-inflammatory things and stuff in that plasma, but the argument you should be giving them more antibody, I'm not sure is compelling.
Yeah, I think, this whole question of the antibody that's being detected in the various assays, as well as what might be in patients' CCP, is a practical one as well as immunologically theoretical interesting one. And from my earlier years before I became a blood banker, I did a PhD in immunology, and my focus was with antibody repertoires, in particular with anti-DNA autoantibodies and then anti-red cell autoantibodies, doing some work there. And I've always been interested in the diversification of not just antibody repertoires, but T-cell repertoires and what have you in the immune system. There's no doubt in my mind that this is very complex, and we often simplify these things in discussions, and we have to be careful, I think, about that. I know that when I have my rounds with our residents here, and we're going over the cases, and we have a panel, and the patient has a new antibody there, and the resident will say, well, they got an anti-Kel antibody, for example. And I said, really? Do they really have an anti-Kel antibody? Or do they have anti-Kel antibodies? Because really what we're seeing is a collection of populations of antibodies of differing affinities and probably isotypes, idiotypes, definitely. And that this is what I think gives power to the immune system, immune response in individuals, but it also complicates our diagnostic abilities, whether they be for anti-red cell antibodies, antiviral antibodies, autoantibodies, you name it. But I think we have to keep these things in mind. So when I hear that a patient has turned negative for an antibody, I do have to scratch my head and say, I wonder if really we're not picking up the other antibody populations that may still be there and poised, ready to go. Absence of proof is not proof of absence in that regard. So I try and...
Claudia Montiel, please call operator. Claudia Montiel, please call operator.
Sorry about that. I'm in my office. So I try and keep that perspective with all these things.
Well, and it's true that when we look at our repeat donors and see changes in the anti-nucleoprotein versus spike levels, they don't necessarily move in sync. So a point well taken.
All right. I have another question. It says, since the new EUA for CCP has, quote, hospitalized COVID patients, quote, end quotes, as a criteria, do you think this will increase utilization? And is anyone implementing a screening process for requests as supplies are limited in some areas? Plasma is limited in some areas.
I think there's no doubt that the demand is going to increase. And I think some centers are poised in a good way to address that because they're actively screening. I think, Lou, you said you guys are screening right now?
We're screening what?
You're screening for your donors for SARS-CoV-2 antibodies?
No, we're taking referrals, self-referrals of patients and referrals from hospital systems and from public health departments of people who had a positive PCR.
Oh, okay.
And then we're actually, because our experience is over 95% of those people have antibodies, 28 days and beyond, we make their first donation is done, and then we test them for antibodies. And they're virtually all antibody positive, not all high titers, but and it's very efficient. The donors make a single visit and they're drawn and in the system.
Well, it's too early to see demand, but you think the demand is going to increase? I mean, it seems like it is.
Yeah. As much as people are trying to qualify the irrational exuberance of the announcement, there are a lot of people that think it's now a standard of care.
Yeah.
And I don't think it is. But we've gone from 400 plus units in reserve down to 160, 170 now. And we're very concerned that we're going to be short, but we're ramping as hard as we can.
Well, the other concern I've got is the impact on other studies, because, for example, there's some interesting concepts around, well, maybe IVIG works, and then there are other immunomodulatory, the Regeneron study with the hybridoma. And so to me, if this, the question is, will this really impede recruiting people into some really potentially good trials? I'm afraid of that.
Well, would you let yourself be randomized, Art?
See, that's what I'm saying. Yeah, right, exactly. I don't think so.
I will say, we probably forget that there are facilities that either didn't go through all the paperwork to enroll in the Mayo EAP or didn't have a research facility to try to get any individual INDs. And all of those smaller places can now get convalescent plasma. So I think maybe the demand at places that were already enrolled in the Mayo EAP, maybe that demand won't necessarily go up, but places that couldn't get convalescent plasma before, for sure they'll be asking.
Yeah, I think there were 2,700 facilities approximately in the EAP, and there's over 5,000 non-governmental hospitals in the U.S. So we got probably to half the hospitals that were in the EAP, and we're very concerned. And we've really, in our hospital, said we've given them a list of criteria that drives them to the subgroup in the EAP that appeared to have the most benefit. Whether that'll work, I have no clue.
Yeah, but even under the EAP, when we had our spike, we were getting close to a point of having to sort of triage. And so I think now that, you know, the gate's open, it's triage may become a necessity. That raises an important question, because so the question is, so who, you know, if you were going to triage, which patients would perhaps be most, you know, would it have the most benefit in? And that's an open question. Right?
And that's actually getting questions on that right now. If you're going to do a screening process for your CCP recipients, who's going to, you know, who are you going to give it to first? The ones that are most likely to benefit from it and recover, or the ones that are the sickest? How are you going to make that decision?
Yeah, I personally really struggle with that, because your inclination is to want to give it to the sickest, but the data all suggests that you're better off giving it to the ones early in their course. Yes. But when you've got someone who's who crumped quickly and is now on ECMO, how can you tell them, nah, you're too sick for convalescent plasma? It's just a weird statement to make, you know? So well, I struggle with that personally.
The response of ECMO is bad enough.
Well, that's true.
Yeah. We're actually collaborating with our infectious disease docs. We were in the Mayo EAP. I guess that closes on Monday.
Oh, it's done. No, it's Friday. It was Friday, right?
No, that was for enrolling people. I think on Monday is the last day for transfusion of people that you've enrolled. So we've been meeting, and we're currently grappling with that because we anticipate that there's going to be an increased demand. And I think it'll be, you know, a combination of some of the input from ID, as well as hospitalists, ICU docs, and then the blood bank, especially with regard to resource availability.
Well, if you use ID docs, you'll have the best.
we got a good relationship with the ID guy. Yeah.
So let me ask a bean counter question. So BARDA was covering the cost under the EAP, and I guess I've heard that BARDA still has some funds. So what are the economics of conversion to EUA?
Well, the last that I heard, which was midweek, is there may be BARDA money that will get us past the middle of September, maybe to the end of September, directly reimbursing the collector, not the hospital. Just the hospital doesn't get a bill, and we're working the blood organizations, Red Cross, ABV, and ABC, are working to get that extended in some way, shape, or form with federal money under the EUA. So the hospitals are kept out of it. Whether that'll happen or not, I don't know. But certainly, if it gets rolled into a DRG, it's going to create a lot of difference.
Oh, yeah.
So stay tuned would be my answer.
All right.
Yeah.
Also on the bean counter side, I had heard that the blood centers are not to be billing the hospitals.
Correct.
Okay.
Our money comes directly from BARDA.
Okay.
After collection of the unit. Okay. And we don't bill the hospital. They get a $0 bill is what happens.
I'll take 3 liters.
No, you won't.
All right.
You should come to Houston in May.
Let's see. I've got a couple more questions. This one's, how many potential CCP donors will have received CCP transfusions during their treatment? Has anybody looked at that? Do we know? Is it part of the screening questions? I mean, you ask them if they've been transfused.
Yeah, it's part of the screening questions. Yeah. We have no idea. It hasn't happened yet. We're 200,000 whole blood a year, so we're not a big center. We haven't seen it yet. We haven't had anybody come in that said, I got plasma and now I want to give it back.
All right, and I guess our last question is, because we're getting close to time, or we're at time. Let me pull it up. All right.
Synthesizing red cells.
Well, this one's actually for Lou. This is an easy one, Lou. What is the major emerging pathogen we need to be alerted for in the near future that will be a challenge for us in the blood supply?
Well, my biggest concern is my chronic COVID encephalitis. I haven't had a rational train of thought for weeks. I think it's probably the next arbovirus that we don't know about is high on my list. But the fact that we had COVID come, which was unlike any other pathogen we've ever had to deal with, I think tells us we just have to be alert. We need to be doing the horizon scanning that I hope I described in my talk to try and stay ahead of what might happen. The past 20 years, arboviruses have been a big deal in our concerns, but now COVID tells us it's not just arboviruses. So we have to be alert across the range of pathogens and ask ourselves which ones are likely to be a problem or could be a problem, theoretically.
Quit invading other biomes is what you said.
Yes. Stay home, wear a mask, and wash your hands.
Well, everybody, I'm getting all these messages. I have to close this up. We are going to roll into the round table discussion at 3 o'clock. It's going to be a lot of the same type of questions. We're going to continue this discussion. So please join us. The format will be a little bit different. From what I can understand, I'll be able to bring you in. You can ask the question yourself. If you don't want to ask the question yourself, you can send me a chat message and I'll ask it for you. So we're going to start that here in just a few minutes. But I want to thank all the experts. Y'all have been wonderful. Thank you very much for participating. And to the membership, thank you for sending some great questions. And we'll see you here in just a few minutes. Thank you.
Thank you, guys.
Bye. Thank you.
Bye-bye. Bye, everybody.